So hi, everybody. My name is Mason Meisner, and I'm the newish Northern Chapter President. And before I hand it over to Mallory, I'm just going to go over a few updates, and I don't have anything crazy. I sent you guys an email at the beginning of the month, probably like a week after the month started, just on some chapter updates and upcoming events. So today, if anybody went to it, it was the Xeriscape Demo Garden Cleanup. And I think these are happening. There's multiple events for this. So today was one, and then there's one on the 25th. And this event is co-hosted, or it's hosted through the city of Fort Collins. And you can, and to sign up, you have to register through through the city's Engage website. Let's see what else is going on. The 17th annual Starter, Barter, and Plant Sale starts May 4th. And for this, essentially, I don't know if Maggie's still here, so she can kind of correct me. But we partner with the city. Sorry, guys, I'm going to read this. The city of Fort Collins has a new Xeriscape educational opportunities page with lots of free upcoming events and classes. Okay, I read that wrong. Okay, apart from that, those are my only updates. Okay, all right, Mallory, I'm gonna hand it over to you. I'm gonna introduce, so this is Mallory. She's a PhD student at CSU, and she has a wonderful presentation for us tonight on delinquent and late night pollinators. Thanks for being here, Mallory.
Thanks for having me. So I'm going to talk about the delinquent late night pollinators. It's a story about nectar bats, agaves, and hummingbird feeders. Just going to mess with my screen a bit to make sure it all looks good. Cool, got my laser pointer. So I'm going to be going, or for the talk today, kind of the layout will be, I'll introduce who I am, where I'm from, share some fun bat facts, because I'm aware that I'm a bat biologist talking to a native plant society. So I want to just go over some of the basics. I'm going to talk about nectar bats and how they evolved with their plants and the special relationships they have. I'll specifically talk about agave and bat mutualism. And if I have time, which I hope I will, I'll go over some of my preliminary findings and research that I'm doing down in New Mexico. And hopefully there'll be time for questions. So I'm Mallory, as Mason introduced. I use she, her pronouns. I'm in the 5th year of my PhD at Colorado State University, and I'm a PhD candidate. And the title of my dissertation is Assessing Drivers of Long-Nosed Bats at the northern distribution of the range. So drivers, sorry, and I did not read what it says, but it's the same thing. So I was like, trying to see why they are in the area they are when they are there, and why do they leave when they leave. I'm part of Catherine Stoner's lab in the Department of Fish, Wildlife, and Conservation Biology at CSU. And I want to recognize that the research that has been done in Catherine's lab has been going on since 2016 on this particular site that I'm at. She started working with long-nosed bats back then while working at New Mexico State University, and with her in the Department of Fish, Wildlife, and Conservation Ecology. Her master's student Scarlett Sellers and Rachel Burke worked on the initial stages of this project. And when she moved to CSU, Teresa Laverty joined her lab and started working on there. She's now at New Mexico State University as well. And now we're continuing this research at University of Arizona. And I'd also like to thank all of my past field crews who have helped me throughout the last 4 field seasons. So some fun bat facts. And I can't see people. I usually do this kind of talk in person, so it's a bit of a bummer. But I'm going to tell you a bit about bats. Like, what is a bat? Bats are mammals. They have been on this earth for more than 50 million years. They're the 2nd largest order of mammals. So there's approximately 22% of mammals are bats. They can be found on 6 continents, excluding the poles. They're the only mammal that can truly fly. And some bats hibernate and some bats migrate. Bats are in the order Chiroptera, which chiro, like kind of like chiropractor, it's Greek for hand, and ptera is Greek for wing, and you might recognize this from pterodactyl. So when you put it together, chiroptera is winged hand. And to like kind of explain the hand part, I have this diagram to show you. So a bat's wing is actually just modified fingers. So right here, that little blue part is its thumb, which is its little claw right there. And the fingers are here. So this is thumb, second finger, third finger, fourth finger, fifth finger. And then there's just a membrane between each finger. So for our arm, forearm, and it comes down all the way to their ankle, from their pinky finger to the ankle of, that's their foot. So their bats are basically just have like really big hands with skin between them, and that's what allows them to fly. So there's two suborders of bats. There's the microbats, or Microchiroptera, which are the most common bat species, or the only bats that are found in the US are microbats. And then megabats, you might be familiar with them. They're often referred to as flying foxes, and those are Megachiroptera. They're mostly fruit-eating bats. The microbats generally use echolocation to find their food, while the megabats use sight and smell. So echolocation, a lot of us are probably familiar with this. It's very similar to like Marco Polo, yelling out. So the bats will emit ultrasonic sound through their mouth, and the sound waves, when they hit an insect, will bounce back at them. And then they have really large ears, or some bats have a modified nose or mouth that they're able to receive those waves, and or they're extra sensitive to feeling those waves, and they're able to locate where the object is that they hit, and they'll change the pulse. So usually they'll do a search call where it's just like chirp, 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 and then when one of those bounce back at them, they'll start chirping faster. So like chirp, 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 until they get the bug, and then they stop. So bats live in a variety of environments or habitats. So some bats live in cacti. I think this one is in a cactus. I think those are vampire bats in a cactus. And some bats live under palm leaves. Some bats roost in trees. Bat I study is here. It roosts in a cave in large colonies. Some will roost in logs that are rotted out or in crevices. A lot of bats roost in the bark of trees as well, and those are very common out here in Colorado. I'm not going to go through all of this for the sake of time, but bats hibernate or go into torpor. So they have multiple strategies, or either hibernate or migrate when it gets cold, or they're resident, so they stay year round. I guess hibernating is resident too, but some of them migrate to their hibernaculums, or where they hibernate. And when bats hibernate, they will reduce their metabolic rate, heart rate, and respiratory rate. So they go from 200, 300 beats per minute, because they have a really high heart rate generally, but they can decrease it all the way down to 10 beats per minute. So this allows them to extend periods of time without eating food or drinking water that they can extend. So bats will still come and wake up throughout the winter and get some water and try to find something to eat. Just for a few hours or a few moments sometimes throughout the winter. So they're not asleep for months on end, kind of like bears. Many species of bats move between summer and winter habitats for migration, and some search for more abundant food sources. Okay, so sorry. I got in my head about this one. But many species of bats move between summer and winter habitats. Some of these habitats are short distance movements, and for a few bats, they have longer distance movements. A lot of the time, this is to be in search of more abundant food resources, or warmer locations, or places with specific resources they need to take advantage of for, during periods of mating or reproduction or other things. Some seek ideal habitat for hibernating in winter or raising young in the summer. Most species that roost in trees, such as hoary bats, migrate south for the winter when insects become scarce. As you could imagine, trees probably don't stabilize temperature as well as a cave would. So bats have a variety of diets. They're super diverse. Bats eat insects, nectar, pollen, fruit, small mammals and birds, fish, and even blood. And so this variety of adaptations of physiological, morphological, or behavioral characteristics allow the animals to live in various environments. That right here is the fringe-lipped bat. It's a species of bat that eats frogs. These bats are also known as frog-eating bats, and they're found in the tropical lowland forests of Central and South America. A lot of folks study these bats out in Panama, and it's really cool. They will they use echolocation to detect the ripples in the water, and that's how they find the frogs. And there's also bats that find fish that way too, with the ripples. And these bats, the fringe-lipped bats, can tell the difference between frog calls, so they can tell which frogs are poisonous and which ones aren't, just based off their calls. So why are bats important? Bats provide some pretty important ecosystem services. They eat insects that destroy crops or transmit disease. They disperse seeds to help the forest grow by eating fruits and pooping it out as they fly around. And they pollinate many plants, as they'll get into in a little bit. But many bats are in danger of extinction. The ongoing destruction of natural habitats, hunting and persecution for sport and meat, the growing use of wind turbine energy, the proliferation of harmful myths, and changing climate are all threatening bats. So just to explain a few of these, but for wind turbines, I guess, might be the most relevant since we're starting to put up a lot of those more. But bats and birds are both threatened by wind turbines. I'm not anti-wind turbine, but I think this is something that we should all be aware of. So if you think about the tip of a wind turbine, or a blade of wind turbine, it's very large. And when you look at them, the rotating at a speed that looks not super fast, but if you think about how large it is and how fast it would take to make a whole rotation, the speeds reach or go or fast enough that it can actually change the air pressure of the air or the space surrounding the blades. So there's this extreme pressure change from like right in front of the blades to within a few inches of it. So when a bat or a bird approaches a wind turbine blade, they don't even have to touch it. They just enter this area of pressure change and their lungs explode, kind of like if you were came up too fast from scuba diving or something depressurized while you're in it. And we call that barotrauma. And unfortunately, wind turbines are set up in areas of best wind, which makes sense. But those are also overlap with historical migration routes for these species, migratory species. And then even more unfortunately, through our monitoring efforts and research, that researchers have found that bats are actually attracted to the wind turbines. There's little thermal camera videos of the bat like flying by. It actually passes the wind turbine, like avoids it, and then comes back and checks it out. And it's like looking at it and then just drops down to the ground because it gets too close. So something about wind turbines, they must emit an ultrasonic sound that attracts bats towards them. And it's unfortunate and should be taken into consideration. Other things that are killing off bats. So that's killing off or threatening the migratory bats. But for bats that hibernate, there's this fungus that was brought over from Europe back in 2008 called PD, or pseudodestructans. And we call it the disease that it gives the bats, or sickness, is white nose syndrome. So what happens is this fungus grows on the bats and they wake up multiple times throughout their period of hibernation. And since their metabolic rate heart rate is raising every time they wake up. They end up using or burning through more of their reserves than they were prepared to do. So then these bats end up dying of dehydration or starvation throughout the winter. Okay, so now a few more positive, or a bit positive, away from the negative. So usually I like to do it back and forth, but since it's online, that didn't seem possible. So I just wanted to share a few other bat facts with y'all. So there's a common misconception that all bats have rabies or that they have a higher instance of rabies, and this is a myth. Transmission of this disease when encountering a wild animal is a common concern. However, it's now thought that less than 1% of bats are rabid. And a lot of this, if you are in a room with a bat or if you get bit by a bat, you should definitely get tested for rabies, because a lot of times you can't even feel that you got bit. And that's the biggest threat that bats have. Usually you don't come into contact with bats, and if you do, they might be a sick bat. But just because there's a bat acting totally normal around you doesn't mean it's rabid, but always worth getting vaccinated for. Another fun bat fact is that the world's smallest mammal is a bat. The bumblebee bat is less than 3 inches long. It's so tiny. That's an adult. This is not actual size because I don't know how big your screen is. The world's largest bat, in contrast, is the giant golden crown flying fox. And if you could guess, I mean, don't guess because we're in a video chat, but just think to yourself how long you think its wingspan is. And it's 6 feet, which is taller than me. It's insane. It's not very tall, so they're not super scary, but its wingspan's longer than mine. I think these are found in Southeast Asia. And the fastest bat in the world is the Mexican free-tailed bat, which we do have here. And they can fly up to 100 miles per hour. And there's some researchers at the university who are currently trying to find the highest elevation maternity roost for this species over in Pingree Park. Or not Pingree Park, Mountain Campus. I don't know, maybe that's Pingree Park. I've never been there. But just up in the Pooter. So that's really cool. You can fly over 100 miles per hour. Bats hold the record for the oldest small mammal ever recorded. And this one was found in the wild. So it's a tiny bat from Siberia. It's called a Brandt's myotis. Holds the world record by living over 41 years in the wild. So it was captured, then released, tagged and released, and then they recaptured it 41 years later. So it was an adult when it was captured too, so it's probably older than 41. Well, it's definitely older than 41, but we don't know how much. And the reason I'm here is because bats are important pollinators. Bats pollinate 528 plants, and including 300 species of fruit. And these are common fruits that you may have not realized, or at least I didn't realize that bats pollinated until I started working with them. So like mangoes, bananas, cacao, peaches, cloves, and even agave, which is used to make tequila and other agave products. So in Colorado, there are 19 species of bats, which I have all listed, or pictures of almost every single one here, but I was missing one and couldn't figure out which one it was. And they're all the Microchiroptera, or microbats, or insectivorous ones. And then in New Mexico, where I predominantly work, there's at least 30 species of bats. When I look at the species list, I'm always like, oh, well, there's one missing that I have caught there. So I think at least 30. And I like to highlight a few of my favorites that occur in both Colorado and New Mexico. So one really cool bat is the pallet bat. They go for ground-dwelling insects and arthropods that other bats ignore. And arthropods are the animals or insects without a backbone that have an exoskeleton like crickets, grasshoppers, and beetles. Pallet bats will also eat spiders, cicadas, scorpions, and centipedes. There's this BBC Hunt episode where for the desert, where it zooms in on a pallet bat in the Sonoran Desert, I think, and it's crawling on its little elbows, tracking down a scorpion. And they seem to be immune to the venom because they can get stung and they don't really react. They can also eat desert centipedes, which are poisonous. So it's very surprising. Oh, there. I have it. So they hunt for food on the ground. They target ground-dwelling insects and arthropods. Then there's the Brazilian free-tailed bat, the Mexican free-tailed bat, the same one that I was just talking about with the maternity roost here. And there's, in New Mexico, there's over 400,000 hungry Brazilian free-tailed bats that emerge every night to feed on insects out at Carlsbad Caverns. So there's other large emergencies where folks claim there to be upwards of close to 1 million bats emerging at Bracken Caves in southeastern Texas. And there's a lot that come out of Union Bridge over in Austin, Texas. But the impact that 400,000 individuals have emerging every night to feed on the local ecosystem is very large. You think about, they are a food source to predators in the area, to birds, to snakes, to skunks. And they also eat massive amounts of insects in areas where we have heavy agriculture. So they're saving us from the use of pesticides and other things that would eventually work their way into our water system. So thank you, bats. And then I'd like to highlight the 2 nectar bats that I currently study. Well, 3, because this one's 2. There's 2 that look like that. And these only occur in New Mexico, and they do not occur in Colorado, but they come very close. So I studied the long-nosed bats and the Mexican long-tongued bat. And these are both important nocturnal pollinators, and the long-nosed bat is an important migratory pollinator. So get into that. But first I'll go over nectar bats. Nectar feeding bats only make up about 5% of all the bat species. For microbats, the Philostomidae family, or leaf-nosed bats, hold the majority, or all of the nectar bats for microbats, and majority of nectar bats overall. They occur in 2 divergent neotropical subfamilies. And so one of the families has 20 species, the other one has 36, but that means they evolve these traits independently. So when we look back at lineage, they come from insects, or insectivorous bats are what they're most closely related to. And then with the megabats, so the Pteroptidae family, or the old world flying foxes, has nectar specialists in 3 of their subfamilies with predominantly ferruginous lineages. So this one seems where bats that eat fruit just also start to eat nectar, which seems to make a bit more sense. And so even though 5% of bats are nectar specialists, as I said earlier, 528 species of angiosperms are pollinated by nectar bats and depend on nectar bats as primary pollinators. And these plants tend to exhibit bat pollination syndrome, or something that I like to call chiropterophilia. So chiropterophilus flowers, or bat-loving flowers, are generally night-living flowers that produce nectar in the evening, white or light-colored petals. The petals point up towards the sky at night, so the moonlight illuminates the flowers so the bats can find them. And the flowers emit potent scents to attract bats and often have elongated trumpet-like shapes. So kind of similar to when you think of flowers that a hummingbird might feed on. The plants generally stand out in the landscape, making it easily navigable for bats. So as you can see, a lot of these plants, well, this one's pointing down, so that doesn't make sense. but a lot of the flowers point straight up and they're light colored and they're very long. So it forces the bat to put their face in it. So when bats visit the Chiroptilus flowers, they stick their heads into the flowers to access the nectar. And inadvertently, while doing this, they transfer pollen onto their fur. And as they go to each subsequent flower throughout the night, they transfer pollen to those. So they can move pollen from a plant. And some of these bats will forage like over 100 kilometers a night before returning to their roost. And that's just a round trip travel. And one-way travel can be even further. And by doing this, they're able to take pollen from a plant that doesn't have wind dispersed pollen. So the pollen wouldn't move very far on its own, and seeds aren't wind dispersed, they just fall. They'll take pollen from that plant and move it 100 kilometers to another plant, and therefore spreading genetic diversity. So this results in a mutualistic relationship where the plants are pollinated as the bats feed on nectar. Plants aren't just pollinated, they're pollinated at a higher rate with genetically diverse pollen. So nectar bats have unique morphology. The bats share physical traits that help them forage for food or forage on or feed on and access the nectar of tubular flowers. So commonly shared traits of nectar bats are the elongated rostrum, so space, and a longer tongue. And they use this to reach nectar deep in the tubular flowers. The reduction in dentition, or their teeth, so less teeth and or a gap between their lower incisors, make a space for the tongue to move in and out and be able to move nectar in faster. So the tongue even has a specialized pumping mechanism. That's why they need this gap. So the tongue just sticks out the mouth, and their mouth is basically closed while doing this. And the tongue can pump the water into it by the way that it moves, rapid consumption of nectar to minimize the time and energy spent hovering. So if you think about like how your cat, when they're drinking, it's the water's kind of moving in and they're not kind of lapping it. I don't know if you ever looked close at that. And their ability for hover flight and their adaptive metabolism is unique to nectar bats. Their hovering is energetically expensive, but common in nectarivores, and it's necessary for them to feed for most of these flowers because there's nowhere to perch. So nectar-feeding bats exhibit specialized behavioral and sensory adaptations to locate their food sources. So unlike insectivorous bats, which use echolocation, nectar bats use sight and smell as their main sensory cues while foraging. They first locate potential foraging grounds using sight by spotting the silhouettes of flowering stalks or cacti. You see this, the cactus is very dominant on the landscape and they tend to flower at the top. So this is a saguaro, and that's a saguaro flower. So they're at the top in a crown pointing up at the moon. Second, they locate fresh flowers using smell. So once they find all the standing things, they don't know which ones are fresh, and sometimes it's not super bright, like you don't have a camera light lighting it up like this. So they'll smell around to catch the scent of a really fresh flower. So New World Chiroptilus plants contain scents such as dimethyl sulfate that signal the presence of fresh nectar to bats. Additionally, philostomid bats use echolocation. So they emit calls as they approach potential food sources, and specialized petal shapes distinctly reflect the echoes when open. And I have the picture of the saguaro flower here because it's really unique in the disc shape that it has. It's been studied where once it opens, it will like vibrate back and send. When the bats echolocate, they can hear the unique call or unique echo of the saguaro flower, where it doesn't do that when it's after it's wilted or before it's open. So back to the bats that I study now after doing that. So I study the lesser long-nosed bat and the Mexican long-nosed bat. They're both species of conservation concern. The lesser long-nosed bat, or Leptonicterus yerba was removed from the endangered species list in 2018. And they were the first bat to ever be removed from the endangered species list and the only bat to have been removed so far. And then there's the Mexican longest bat, Leptonictus nivalis, and it's currently listed as a federally endangered species. There's also Corionicturus mexicana, or the Mexican long-tongued bat. It's a different genus, but similar bat. It was a candidate for listing as an endangered species in 1991, but in 94 it was deemed data deficient. So part of my postdoc when I moved to University of Arizona will be studying this bat more closely. So how do you tell these two bats apart is a question I commonly get, maybe something that might not concern folks on this call as much, but I'm still gonna tell you because I think it's cool. Leptonictus versus Corionictus, so they have a different tail membrane, and we like to call this pants versus skirt. And you can't tell the two Leptonictus apart at all because you have to like measure basically this joint of their pinky, so to see if that bone is slightly larger or not, and then there's overlap, so it's even hard to tell apart when in hand. But as you can see here, Leptonictus has a pants, so there's a gap between its feet or its legs, so it doesn't have a tail membrane there, where Corionictus does have a tail membrane. It's connected. They also have different snouts. I like to think of the Corionictus as looking like a horse, where the Leptonictus looks more like a dog. Lepto has a shorter and broader snout versus Corio, which is longer and narrow. They also have different body size, which is hard to tell from a photo because, you know, it's nighttime, you have nothing to gauge how far away they are. But Leptonictus is 20 to 35 grams, where Corionictus is 10 to 25 grams. And they have very different life histories. Leptonictus is a long distance migratory bat, where Corios are not. So the Leptonictus weigh more because they have more muscle mass. So Leptonictus is known to roost deep into caves and mines in large colonies of 1,000+ individuals, where Corionictus is known to roost in the entrance of caves and mines or in crevices in small groups of 1 to 13 individuals. Leptonictus can be found in the Gila National Forest, is one of the most northern distributions of the species range. So that's down in southern New Mexico. And it occurs in the US in Arizona, New Mexico, and Texas, where Corios are found in the US can be found in Nevada, New Mexico, Texas, California, and Arizona. So they have a larger distribution. I'm going to tell you a bit about agave and bat mutualism. So our research focuses on these two bats. They're important migratory pollinators and seed dispersers. They provide critical ecosystem services, contributing to healthy soils, diverse vegetation communities, and sustainable economic benefits for communities through ecotourism and pollination of crops such as agave to produce tequila and mezcal. So long-nosed bats are migratory bats characterized by a resident and migratory subpopulation. So resident subpopulation remains down in southern Mexico or southern central Mexico to mate and give birth, while the migratory subpopulation consisting of mostly pregnant females travel approximately 1,200 kilometers north each spring to give birth at maternity roosts in northern Mexico and southwestern United States. So come up here, give birth over here, come hang out with me in New Mexico for a bit, and then come back south. And there's mixing between these, or genetic mixing. So they're not distinct populations, subpopulations. When they migrate north, they follow a hypothesized nectar corridor that was described as a spatial temporal migration corridor with stopover forage and roost habitats that promotes seasonal dietary specialization and assumes a mutualistic relationship between the pollinators and their plant food source. So the pregnant females will follow their food sources as they flower up north, and then follow over here as more flowers, and then come back down and follow another flowering path. So I think that's kind of pretty. The arrival. So I work over here in the northern region of the Chihuahuan Desert. The arrival of bats to this region is thought to coincide with regional flowering of the agave species, which is the only known plant nectar food source used by long-nosed bats in New Mexico. So the agave species is in the Asparagiaceae family, and it does kind of look like asparagus when the stalk is first emerging. It takes 8 to 30 years to fully mature and flower. They're monocropic, which means it flowers only once in their lifetime, and after flowering produces fruit and dies. So it's a long-term investment into flowering. This is mostly because of the bats. Agave have a basal rosette, a paniculate flowering stalk, so a straight stalk with arms that come out, has lateral branching with a distinct umbel at the end of each branch. And flowers, there's about 150 cream-colored flowers that point upward and release nectar shortly after nightfall. And the umbels begin blooming on the bottom branches and proceed upwards. And this can take anywhere from 1 week to fully flower to 8 weeks to even longer, depending on the species. So here I have some labeled images. So this is one bud out of like the 150 per umbel that I picked off or found on the ground. This is what it looks like in the bud stage. The flower becomes staminate, then pistillate fruit. And that's a fruit that was actually successfully pollinated. So it's protandrous, which means the male reproductive organs come to maturity before the females. So day 1 through 4, the stamens and anthers develop and release pollen. And then day 5 through 10, the stigma emerges and becomes receptive. At my study site, we have 2 agave species. We have agave palmeri and agave perii. So agave palmeri, or Palmer's century plant, has a basal rosette that's 1 to 2.5 meters wide, so it's very large. Sometimes I'll even have like a 3.5 meter or 4 meter basal rosette, kind of like in this picture here, where it's the same height as my field text. The stalk can be anywhere from 4 to 8.5 meters tall. It has lateral branching, 11 to 30 arms. Approximately 1 to 3 umbels open at a time, and blooming can last up to 8 weeks. And for the agave perii, it's the higher elevation agave plant that grows out where I work, so it kind of takes a lot of work to get to one. They have a basal rosette of 0.4 to 1.5 meters wide. The stalk can be anywhere from 2 to 4 meters tall. It has lateral branching, 11 to 30 arms, and all umbels open within a week, and blooming can last up to 2 weeks. So at higher elevations, agave perii can be found growing in sparse patches near mountain peaks in the boot heel, and can be found abundantly further north. At the northern distribution of these bats range in the Gila. But most of the diet studies for the bat I study exclude agave perii because it's so hard to access them, to study them, that data has been absent for it. So I've been working extra hard to get to those. Is that a different slide? Oh well. So due to the absence of flowering cacti in New Mexico and eastern Arizona, the agave plant serves as the only known nectar plant food source for the nectivorous bats. So the agave species exhibits the chirolithic pollination syndrome that I discussed earlier. They exhibit morphological features that are congruent with the chirolithic pollination syndrome. So their floral shape and size, so the upward facing yellow petals, the nocturnal nectar production. They start producing nectar like right at sunset, usually around 9, actually, like a little bit after. And I can see the bats emerge. The nectivorous bats tend to emerge about a half hour to an hour later than the insectivorous bats. I think it's because the agave hasn't started producing nectar yet, so they have no reason to leave. Nectar quality is high. It contains proteins and amino acids. And the high sugar concentration, it ranges from 18 to 26%, which is what the bats are looking for. And the nectar bat's characteristics, so long-nosed bats, they have the elongated rostrum, they have a long tongue, and they have an accelerated metabolism based on nectar sugar concentration of 5 to 29%. So they overlap the range of the agave sugar concentration. So we want to know why the bats are there, because sometimes the bats aren't in New Mexico when we think they should be. So Rachel Berg, the former master student of Dr. Catherine Stoner, tested the spatial temporal nature of the nectar corridor hypothesis with independent data and determined that species richness of food plants is a major driver for migration and distribution for all 3 nectar bats found in the US. So this map is a seasonal distribution model of Leptonectar's yerba buena. And the map shows suitability for lesser long-nosed bats based off of nectar food availability. So winter, top left, spring, right, summer, and then fall. And the green is low suitability, where red is higher suitability. So the map, or as shown here, the distributions of these bats were the most uncertain during migration periods in the spring and fall. You see how like the yellow is very spread out in both these areas. It's not so concentrated like it is in the winter. And the red's a bit more concentrated in the summer, which suggests that factors other than species richness of food plants may influence migration patterns during that time. And during that time, these bats are in New Mexico and Arizona, as you can see here. And another part of the agave bat mutualism that recently came out is a recent study by Trejo Salazar constructed this 3 co-presence matrices between agave and 2 long-nosed bats to explore the possible dynamics of their biotic interactions. And they modeled the agave leptonectar's coincidence, like the coincidence that these 2 things would be here at the same time in the way that they are. So in its space and in its time components, and inferred the historical presence of 2 agave leptonectar's interaction corridors. So these 2 migration corridors that we had shown. So the one going to Arizona, and this one going to Big Bend National Park, which is where a Nivalis roost is. So they infer the interactions of those corridors with the stability from the last glacial maximum to present the agave leptonectar's interaction refuge in areas of the sorry, I can't say this right, but the Metzalhan Valley, which is in near Mexico City, or Techucan Valley is near Mexico City, but these are both very close. And they found that paleontological, geological, and molecular data suggested that these interactions may have remained active for the last 2.5 million years, which is amazing. So some of the researchers down in Mexico, the ones who were working on this, like to say that the bats domesticated the agave so that we could make tequila, because the agave industry is very large down in Mexico. But it's cool, 2.5 million years. So to summarize the agave and nectar bat mutualism, the chirolithic pollination syndrome, the nectar bat's anatomical and physiological characteristics, the geographical congruence of the historical range distributions overlapping, and the temporary correspondence between agave flowering and the nectar bat migration timing all indicate agave and nectar bat mutualism. Indicate a coevolution mutualistic relationship with the nectar bats that pollinate it. And the loss of the food plants along the long-nosed bat migration corridor could jeopardize both the agave and the bat population. So that's where my research comes in. So I'm just going to do a time check. Do I have like, would another 20 minutes be okay? I don't really remember what time I started.
Yeah, that should be fine. It goes to 7:30, but if people want to hop off, they can hop off early, and if they want to stay on, they can stay on.
If it goes to 7:30, I'll go faster. It's okay. I could do this in less than 10 minutes. But the leptonicture species conservation needs, so they're what these bats need for survival and future sustainability is the presence of suitable roost sites and availability of forage resources. So safe caves to roost in, and they need their nectar corridor to be not fragmented, so continuous. So however, climate change, wind farm development, and agricultural activities such as livestock grazing and agave farming to produce tequila have threatened the availability of foraging resources and connectivity of the nectar corridor. So just to like quickly go over the agave farming thing, when agave is grown for the production of tequila, it they grow to the point of full maturity where the flowering stalk shoots up. But once the flowering stalk shoots up, they cut it off so all the sugars go back into the heart. And then they harvest it 2 weeks later or something. So all those agave that you see in that field there will never go to flower. There is, however, researchers have started working with agave producers to make bat-friendly tequila. And they've done some calculations and found that if you let 5% of plants per hectare to go to flower, it would theoretically produce enough nectar to support the migratory corridor because one agave produces so much. So look for bat-friendly mezcal and tequila. Also, unfortunately, there's been a recent study that predicted a 75% reduction in suitable habitat for agave within the distribution of Leptonicterus nivalis. So the blue here is nivalis. Suitable habitat, the light green is agave suitable habitat, and the dark green is where they both overlap. As you can see, the dark green is like the full nectar corridor, goes all the way to southern Arizona, New Mexico, and over here goes to Texas. But then, however, by 2070, using the most conservative RCP value of 4.5, there's a 75% reduction in suitable habitat. And as you can see, the corridor, that dark green is no longer going to New Mexico, Arizona. It's no longer going all the way to Texas. It's fragmented. So given changing weather and climate may affect the timing and phenology of plant reproduction, it's possible that a phenological mismatch may occur between the presence of long-nosed bats in the US and the timing of agave food availability, which also threatens the future viability of the species. So it could happen even sooner. And there's potentially already a phenological mismatch happening between the bats and agave nectar availability. But there's evidence suggesting that there is potential diet plasticity that already exists. So the agave bloom in New Mexico, typically in early July or early August. Bats tend to start arriving early in June and depart by late October. This suggests that fewer nectar resources may be available to bats in late spring or early fall and indicates a change in resource use between seasons. So here's a little map that I did for this pit tag study I'm a part of. So all these locations on the map, we're currently monitoring caves or known roost sites for tag locations. And then the highlighted sites are where I work. So here's a little zoom in on my highlighted sites. So I work at a cave in the Big Hatchet Mountains there. It's the largest known roost site of long-nosed bats in New Mexico. And then all these starred sites are ones that I monitor bats at hummingbird feeders. So like I said, my main roost site that I study is in the Big Hatchet Mountains, which is in the BLM wilderness study area. This is the cave that we work at. The Hatchet Mountain roost is believed to be a late summer transition roost where long-nosed bats, mostly female or young, stay prior to their migration south. We collect fecal samples from this cave, so we examine the diets of individual long-nosed bats using fecal samples and pollen samples collected from captured individuals, and fecal samples collected weekly from a plastic ground sheet secured to the ground. A subset of fecal samples are sent to Pisces Molecular Lab in Boulder, Colorado for genetic analysis to both confirm the bat host species and identify plant and insect species in their diet. And then we're also going to be analyzing pollen and a subset of fecal samples to try to use microscopic techniques. So this is us collecting fecal samples. My technician Kennedy made this slide. There she is getting poops. There she is rappelling down to the cave. It's very technical to get down there because we want to be safe. And some just preliminary results. So we hypothesized that as agave nectar availability decreased on the landscape, that insect consumption would increase. And our results from the first 2 years is what I have here, are kind of going along with that. So in periods of low agave availability, insect consumption is increasing. So this black line is the fecal samples, like the percentage of fecal samples that we had tested that had arthropods in it. And this green line, or green part back here, is the relative agave availability. So the number of agaves that we surveyed throughout each season and what percentage of those agaves were currently flowering at that time, like had fresh flowers on them. So if we compare these 2%, the darker color, so the dark green black is 2020, the light green and the gray is 2021. We could see that there is kind of a pattern here. And I'm hoping that when I add in my 2022 and 2023 samples, I'll be able to run models that will also see the same thing. And overall, throughout 2019, 2022, 81.5% of our samples that we tested contained insects, which doesn't seem that surprising because people think of bats with insects all the time, but these bats are thought to be nectar obligates. And all past studies done outside of our lab have had very low amounts of insects in the diet. Ours might be higher because of DNA, because we use DNA metabarcoding methods to test our samples. However, Scarlett Sellers, the previous master student, collected samples at the same site, and just with her microscopic methods, she found that 49% of the samples contained insects. So we do think that they're consuming larger amounts of insects at the northern distribution of the range. But then to further examine it, and because all those other times they thought, oh, well, it was probably incidental consumption of insects, like while they were feeding on the flower, they accidentally ate some bugs, or while they're just foraging, bugs landed on them, or while they're in the cave, mites are on them, and they just consume them while grooming or incidentally. But I broke to like further examine that and to see if is it incidental consumption or intentional. I categorized the insects into aquatic insects, which are ones that live every life stage in the water. Insects that are associated with flowers, insects that are ground dwelling, insects that are found on bats or in the cave, and then just an other because it was at too high of a taxonomic index or classification for me to group. And for aquatic arthropods, ones that we found that spend every stage of life in their water, I found aquatic Caloptera, shore flies, fish flies, freshwater pelagic copiopods, freshwater ostracods, and water mites. So this kind of indicates intentional consumption or the use of water troughs, and they're consuming these while drinking. But it seems that all the aquatic insects were consumed in 2020, which was an extreme drought year. So it's interesting, indicates kind of like that they might be using these water troughs or more often during periods of drought. Then for ground dwelling insects, which couldn't incidentally be consumed. I don't know how they could incidentally be consumed. If anyone has any ideas, please tell me. But one of ours, all of that was a desert centipede, and those are venomous, so that's insane. We found hide beetles, green lacewings, brown lacewings, net-winged insects, grasshoppers, common desert centipede, and spiders. So these arthropods are either ground-dwelling or flying arthropods that are not associated with flowers, and therefore have a low chance of being consumed. So in summary, arthropods make up a significant part of the diet of long-nosed bats in this region. Consumption of arthropods occurs at a higher rate throughout occupancy. Detection of aquatic, ground-dwelling, net-winged arthropods suggests intentional consumption, and presence of insect DNA without pollen DNA indicates intentional consumption. I guess I didn't really cover that part. Yeah. So we also have mistnet bats. We capture them. We set up mistnets near the cave entrance. or near hummingbird feeders or water sources or other roost sites. And we wait for the bats to get caught in the net and remove the bat from net. Kennedy made this slide too. Thank you, Kennedy. I don't know if I can get this to play. Oh, I can't. Well, that was a video of me collecting pollen, but it's not working. But we don't really have the time anyway. So when we get the bat in hand, we determine sex, age, and reproductive status. So the epiphysis, we see if that's fused. We place the bat in a bag and measure its weight. Measure forearm, we collect ectoparasites, and we collect fecal samples if the bat poops, and those are gold. We also pit tag the bats. This is a pit tag gun. And we pit tag the bats with these things. They're kind of like the microchip that you would put in your cat or dog, or like an RFID tag. And we have this antenna system set up with all those ones I said where we're monitoring. And when the bats fly through the antenna, it excites the pit tag and it reads its frequency and can mark its unique ID. So we're able to track these bats over long periods of time. So since these tags don't need batteries, it'll last throughout the bat's life. So we've been able to track bats over 8 years so far, and we've only been doing this for 8 years. So it's really effective. We also feed the bat sugar water, and it's a shame this one won't play because it's a really cute video of a bat drinking. But look how long its tongue is, and that's like only a quarter of it, probably. Oh, look, it's drinking. I really like to drink the sugar water. And maybe this will work, the bat being released. Look at those strong arms used to migrate 1,200 kilometers each way, so twice a year. So with those pit tags, we track their movement using our movement study, and we count the number of bats that emerge using a thermal camera. So I have a thermal camera deployed out there on a solar panel. So, and I download data every couple weeks, and we're working on currently getting weekly population counts. So we're looking at the motivations for movement at agave phenology and spatial distribution across the landscape, climatic variables, and long-nosed bat movement and population dynamics. So seeing if flowering agave density, how that affects bat population and the timing. And we non-invasively collect all these things. So it's the thermal camera, it's info on our movement study. I also net for bats at residential sites. So these bats are sticking around longer and leaving later. Like I catch bats in October, which is not, there's no agaves blooming in October, or if there is, it's like there's only a couple. And I think this is due to anthropogenic food sources such as hummingbird feeders. So I'm trying to track these bats at them, and we're making a pit tag reader to track them. And I don't know if this video will play. There it goes. So we create this little pit tag reader, or we use the design by Eli Bridge over at University of Oklahoma, I think. And we're able to, we have a new antenna design that we're going to test this year because the bats clearly don't like that hoop antenna, but it only costs like about $40 to $50 to make. And we want to set these up throughout the US and Mexico at folks' residential sites to be able to track localized movements of these bats, which is going to be really exciting. We use acoustic monitoring, we have weather stations, and I'm trying to use acoustic monitoring to figure out spatial distribution because there's a lot of development for wind energy proposed in the area. And if they do put it in and it's within their migratory corridor, it could decimate the populations of these bats. So yeah, that's it. I think I did that. I was only 2 minutes over. I can stick around if anyone has any questions or wants to see cute pictures of bats.
Yeah, if anybody has any questions, if you guys just want to drop it in the chat, so not everybody's trying to talk at once.
Oh, you're muted, Mason.
I guess I'll ask one. It's probably a silly question. But can all species of bats contract white nose syndrome?
They can all contract it, but they don't all get affected by it. So like my bats don't hibernate, the ones I study, the nectar bats. So even if they were to pick up the fungus, just like how I could get athlete's foot on their little faces, it wouldn't be detrimental to them. But they could, since they do migrate over 1,200 kilometers and stay in multi-species roost, they could be a vector.
Okay, interesting.
Mm-hmm. Oh, there's the chat. Okay, so I see there's a question. Are the bats the only pollinators of agave and cacti, or do night flying moths also play a role? So there's been a lot of studies done on that. So even during the day, there's a bunch of birds and bees and large agave bugs and moths in the evening at agaves. However, when they put bat exclusion cages around the flowers, so only insects can get to it throughout the morning and evening, or AM and PM, they found that the fruit had a much smaller seed set than if the bats were to feed on it. The only time that it's actually a larger seed set is when it's for pallet bats. They've actually been recorded feeding on agave nectar. And since they don't have the modified rostrums, they have like a flat face, they have to like really get aggressive to get in there. So they actually move more pollen. And the same thing, I was going to work on a paper with like procyonids, so ring-tailed cats, coatis, feeding on agaves, and how they might also, since they don't have modified rostrums or faces or anything, they're big, to feed on it, that they potentially also have larger seed sets. So that's something I want to explore in the future. But you lose with the coatis and smaller creatures like moths and insects, you lose that genetic diversity. So you don't have those long distant movement of 100 kilometers of the pollen moving. So it could eventually die from genetic inbreeding. Do you standardize the collection of pollen off the bat? We collect pollen off of every single bat that we catch. We have these little fusion gel tabs that we've created. So we just get a little cube of that and we wipe it off and put it in a tube. We don't have a standardized collection on like how many times we swipe, anything like that, because we're not going to do amounts. We're just trying to look for presence, absence of pollens. But we're very thorough when we're getting it. And if we see pollen, we target it. Did that, I think that answered the question. Let me know if it didn't though. Oh, it's, hey, Holly. Are yucca blooms the wrong orientation for bats? So yucca have, I thought that maybe my bats are feeding on yucca pollen. They actually have these modified sacs that only the moth that co-evolved with it can actually get its little pollen feeder thing in there to feed from. So although I did ended up like collecting some yucca flowers and pulling some pollen out of there, it was like in such a small sac that I see no way that a bat could actually feed from it.